Dark reaction of C4 plants. What is dark reaction? Dark reaction is producing glucose by using energy from light reaction using ADP and NADPH. Dark reaction take place in internal compartment of chloroplast. What are the C4 plants? All use a supplementary method of CO2 uptake which forms a 4-carbon molecule. These plants are called C4 plants. C4 plant adaptations. High daytime temperature. High light intensity. Some examples of C4 plants: corn, sugarcane, sorghum. C4 leaf anatomy. C4 leaf anatomy called as Krantz anatomy. Its inner ring-shaped cells called bundle sheath cells contain starch-rich chloroplasts, mesophyll cells present as the outer ring. Primary function of the Krantz anatomy is provide a site in which CO2 concentrated rubisco. Differences of bundle sheath cell and mesophyll cell. Bundle sheath cells chloroplasts have RUBP carboxylase enzyme. Mesophyll cell chloroplasts haven't RUBP carboxylase enzyme. Bundle sheath cell chloroplasts are lacking grana. Mesophyll cells are rich grana. Bundle sheath cells have large chloroplasts. Mesophyll cells have normal chloroplasts. Mechanism of dark reaction of C4 plants. Now we consider stepwise reactions of dark reaction mechanism. First step in the mesophyll cell. The primary acceptor of CO2 is phosphoenol pyruvic acid. It combined with CO2 presences of phosphenol pyruvate carboxylase from 4-carbon compound oxaloacetic acid. The first stable product is oxaloacetic acid. Oxaloacetic acid is reduced to malic acid using NADPH. This reaction catalyzed by malic dehydrogenase. Then malic acid transported to bundle sheath cell where there are decarboxylated in presence of NADPH and specific malic enzyme to produce pyruvic acid and CO2. Liberated CO2 accepted by RUBP and enter the Calvin cycle. The pyruvic acid formed transported back to mesophyll cells. The phosphoenol pyruvic acid is regenerated by using ADP which produced in light reaction. Significance of dark reaction of C4 plants. C4 plants can absorb CO2 from a low concentration of CO2. It requires more light energy for photosynthesis. Photorespiration does not take place. The plants are better adapted to deserts. C4 plants' photosynthesis efficiency is higher than the C3 plants. Photorespiration does not occur. CO2 acceptor is phosphoenol pyruvate. It is higher efficiency than RUBP in C3 plants. C4 pathway CO2 storing in 2 times. High light intensity photosynthesis efficiency is also high. Difference between C3 and C4 plants. C3 plants leaves haven't Krantz anatomy. C4 plants leaves have Krantz anatomy. C3 plants leaves have one type of chloroplast. C4 plants leaves have 2 types of chloroplasts. C3 plants xylem and phloem surround by bundle sheath cell ring. C4 plants xylem and phloem surround by bundle sheath cell and mesophyll cell rings. C3 plants happen photorespiration. C4 plants not happen photorespiration. C3 cycle first stable result is PGA. C4 plants first stable result is oxaloacetic acid. C3 plants CO2 acceptance only by RUBP. C4 plants CO2 acceptors by RUBP and PEP. Conclusion. Thank you. We are Group B6.
Created using Powtoon.